is ozone. We look at blue light. We've looked at plasma. We also look at antimicrobial fatty acids. A specific area of interest is biocide resistance. We look at how E. coli and Salmonella in particular can become resistant to antimicrobial compounds used in the food industry. And a subset of that research is phenotypic diversity, which I'll explain further down in this talk. So an overview of this of my talk is the interaction between tomatoes and Salmonella. Why is this a big issue in the United States? What is phenotypic diversity? It's something like a diversified portfolio when you are investing in stocks. And then what about metabolomics and how can they create and how can they result in phenotypic diversity? And then we will look at adaptations to stress, some of the genes involved in adaptations to stress. And one of the interesting phenotypes that are formed, which is filamentous cells. We'll also look at biofilms because we've looked at a strange phenomenon with biofilms, and then some of the future focus in our labs. So why tomatoes? And why are they important in the United States? Well, the United States is the second largest producer of tomatoes in the world. For commercial products, the United States is the first largest producer of tomatoes. So it is a high value food product. And as a seeded vegetables, tomatoes are high risk for Salmonella contamination. So in the United States, they use a lot of poultry litter for actually, as an additive to improve the fertility of the soil. And when you use poultry litter in the soil, there's a good chance of Salmonella introduction. Tomatoes are the most commonly implicated vehicle for Salmonella in the United States when it comes to produce. And the major sources of contamination are environmental sources. So the major sources of contamination are environmental sources such as water, soil, and even dust. And Salmonella has been known to persist on not only on the fruit, but also in packing houses where these fruits are actually distributed and transported. So what is an example of a fluctuating environment? So an example of a fluctuating environment could be the surface of a carcass. Suppose you consider yourself a Salmonella and you find yourself on the surface of a chicken or on the surface of a tomato. Once if you're in the environment, you're exposed to sunlight, you're exposed to different stressors, then you're exposed to chlorinated water, you're washed and then packed. There might be some UV radiation. So the environment constantly changes, and that is a fluctuating environment. So foodborne pathogens can experience fluctuating environments in water bodies, food contact surfaces, sanitizer treated surfaces, antimicrobials treated surfaces, and especially produce surfaces like tomatoes. So examples, ponds, and then like for an example, even as a tomato develops, the amount of fatty acids, sugars on the surface of a tomato changes. And if it is contaminated with Salmonella, the Salmonella is exposed to a constantly changing environment. So one of the outcomes of phenotypic diversity is a process called stochastic switching. So what is stochastic switching? So now say suppose you have 10 crore rupees, right? And I ask you to invest your money so that you have enough money that you don't have to work. If you put it only in one industry, there is a good chance that an industry might crash and you might lose all your money. So what you do, you'll invest a little bit in gold, you'll invest a little bit in stocks, you'll put it in FDs, and Salmonella kind of does the same process. So when Salmonella is exposed to stress, there is always a small subpopulation that expresses completely phenotypically different genes than the rest of the subpopulation. This is called phenotypic switching. And the reason for this is, in case there is stress that kills all the other cells, there is always a few that remain, like persister cells. So there is a difference between responsive switching and stochastic switching. Stochastic switching is random. Responsive switching is when you are, say, exposing Salmonella to an antibiotic. Over time, all of it will become resistant to the antibiotic. So it responds to one stress. Whereas here, there are different responses to stressors, and it's random. So why is that important, and how is that associated with tomatoes? Now, surface of a tomato is a rapidly change is a constantly changing environment. So when I was in the University of Maryland, we looked at the fatty acids, sugars on the surface of tomatoes, and we found that different types of tomatoes and also at different stages of development, you would find different fatty acids. We did some research and we found that there was a positive correlation. So Salmonella would grow on the types of tomatoes or cultivars of tomatoes that had more sugars and more protein. But if it had more fatty acids, the growth was not that great. So one thing that we wanted to see was if there was any particular fatty acid on tomatoes that would prevent the growth of Salmonella. So what we did was we did a basic MIC, minimal inhibitory concentration, and we found that salmonella, one particular fatty acid, it was called it's called pelargonic acid or non-anoic acid. It's 9-carbon chain fatty acid, and this fatty acid was extremely microbicidal to Salmonella. So what we noticed was when we would expose it to the fatty acid, we would see cell lysis. And so this is basically a live dead stain. And the green cells are alive. Some of the cells that have, you can see cell lysis, those are dead. What we can also see is some of these cells are extremely long. So these are filamentous cells. So when we would grow these in presence of sublethal concentrations, what we would notice is there would be a dip in population. There would be some adaptation, and then there was a dip. So we wanted to see what was happening. So before we understand what happens, we have to understand the genetic machinery of Salmonella. So one interesting thing is Salmonella have these things called, you have all heard of oprons, but these are called regulons. So regulons are master regulators, and they control a variety of genes. In a lacopron, you will have basically 2 genes. It is associated with lactose, 1 stimuli, whereas here with 1 stimulus, it will end up controlling many genes. So Salmonella has in particular an opron called the 4PQ opron. And the thing that controls the 4PQ opron is magnesium levels. When you have high levels of magnesium, the 4PQ opron is shut down. But when you have micromolar levels of magnesium, the 4PQ opron is turned on. And when it is turned on, it goes on and switches on other genes around it, especially pathogenicity genes and genes for survival. And the reason for this is when a Salmonella is inside a macrophage, there is actually micromolar quantities of magnesium, but outside in the human body, there are millimolar quantities. So it can switch basically based on the amount of magnesium. It can switch into 2 modes, an intracellular mode and an extracellular mode. Why is this important with pelargonic acid? Well, we were able to see if magnesium was important for survival and resistance to pelargonic acid. So you can see that we grew cells in TSB, which is tryptic soy broth, very similar to nutrient medium, and then TSB with 5 millimolar magnesium, and then TSB with just pelargonic acid, and then TSB with pelargonic and 5 millimolar magnesium. What we could see is just the TSB with pelargonic acid was the only one that had a dip. All the others had survival. So basically, magnesium regulation could help in survival against pelargonic acid. So we wanted to see what was happening. How do they survive this fatty acid? How is it that in certain conditions they are able to survive and they are not able to survive? So one thing that we did was we did microscopy. And what we noticed was when exposed to pelargonic acid, and this is Salmonella newport, it's one of the serotypes of Salmonella, around 12 to 16 hours, the cells from Salmonella cell is 2 to 3 microns. From 2 microns, it would become like 12 to almost 50 microns. And around 16 hours, it will almost go up to 50 to 100 microns in length. So the cells from 2 microns would go 50 times their size and increase in length. But this was not a constant phenomenon. Around 20 hours, again, we would see that some of the cells would remain, but many of them would return to their normal size. And by 24 hours, most of the cells would break down to their normal size. So what was happening? Another thing that we noticed was as the cell was breaking down, we could see that we could see the septa forming in between cells. So we were kind of wondering what was happening. And And why were these cells becoming so long? So again, these are Salmonella cells exposed to pelargonic acid. This is basically, to the left is just TSB, no pelargonic acid at 16 hours. This is pelargonic acid at 16 hours. And then this is pelargonic acid with magnesium at 16 hours. So without magnesium, they are forming these really long filaments. So what were these filaments? So we came up with a model. We found out that these filaments are basically a short-term resistance mechanism. So Salmonella forms these filaments by basically stopping cell division. So the cells would divide but not separate. So no septa would form. And so without separating, the cells would get really long. But after a few hours, the septa would start forming, and then the cells would separate from these septa. So what is the use of this? How does this save Salmonella? Well, when Salmonella forms these filaments, we allowed Salmonella to form these filaments and then transferred them one to TSB without any pelargonic acid, and then TSB with pelargonic acid. When we transferred it to TSB without pelargonic acid, we see that the filaments break and there is a shoot or an increase in population. But when the antimicrobial is still there, then the population decreases because the cells separate and still die. So most of the environments, suppose you're using chlorine to sanitize a surface, the effect of chlorine is only going to be there for a few minutes or a maximum for a few hours. So Salmonella can form these filaments, and then after a few hours, when the amount of chlorine reduces, they can break down and actually increase in number. This has not only been found for antimicrobials, it's also been found for antibiotics. So Salmonella typhi can form filamentous cells when someone takes ciprofloxacin for typhoid along with turmeric, because the turmeric will nullify the action of ciprofloxacin, and people have seen filamentous cells form. So this is actually more common than expected. So what did we find out about filamentous cells? They are formed in presence of sublethal concentrations of pelargonic acid. It's a transient morphotype, so it's only there for a short period of time. The formation can result in a spike in population, and filament formation can be subdued by magnesium. So it goes by environmental cues. But then there are 2,500, we have only studied one serotype of Salmonella, which is Salmonella Newport, but there are 2,500 serotypes. So one thing we wanted to see was with these serotypes, with different serotypes, would they have different stress response variations? So we took 3 different types of serotypes. One was Newport, Typhimurium, and then Oranienburg. Oranienburg has been associated with low moisture foods. They have even found it on things like potato chips and all that. Typhimurium is again associated with mice and chicken and all of that. And then Newport, as we said, is with tomatoes. So these were all grown in presence of pelargonic acid. And what we notice, if you look at panel C, F, and I, we can see that they're all forming filamentous cells, but the filamentous cells are slightly different. What we noticed was Newport was forming filaments, but also a little bit of biofilm. Whereas if you look at Typhimurium, they were forming these aggregates. And we didn't know basically what these aggregates were. And then if you look at Oranienburg, they formed really nice long filamentous cells. So we were seeing different stress responses between different serotypes. So one thing that we were interested in is we had this hypothesis that these aggregates were probably floating biofilms. So this became controversial because biofilms are usually attached to a surface. And we were the first kind of people to say that they might actually be floating sometimes in medium when you have antimicrobials. So to confirm that these biofilms, that these aggregates were actually biofilms, we use different types of staining and confocal microscopy. And you can see to the left, these are Salmonella cells. There are filaments and regular cells in a biofilm matrix. And we use a special dye that can actually stain the EPS of biofilms, and it turned out positive. What was interesting, so this is an example of a floating biofilm, right? So when we look at it under the confocal microscope, we could see that there's biofilm material, but also the green ones are green fluorescent protein expressing Salmonella cells. And what we noticed was they are forming these three-dimensional iceberg-like biofilms that are floating in water. So this is a big issue because phenotypic diversity can be seen not only on produce, but it can also be seen in the environment. Anyway, I want to show you an example of how Salmonella can solve its own problems. So if I'm going to switch on this video. So these long cells are filamentous cells, and you see that they are not moving. In between these filamentous cells are small Salmonella cells, and they are trying to find their way out of this maze. You can see that they're, so it's very interesting. They're able to actually learn that if we push in the corner, we might be able to make some space for us. So as you can see, I'm not sure if you can still hear me, but or if you missed out on that, were you able to see the video? Yes. Okay. Sorry, were you able to see the video?
Yes, sir, we are able to see.
Okay, great. So you can see that Salmonella cells were able to escape that. But one interesting thing is, if you go back to the video, you can see that the small cells were motile, whereas the longer cells were not motile. So two different phenotypes, right? So what happens if these long cells need to become motile? So one thing we noticed was, yeah, so the way motility happens in E. coli or Salmonella is there's a certain, there'll be a specific impulse, and then they will change their flagellar rotary mechanism based on the impulse. But what if there is no flagella, because the longer cells are not motile and they don't have a flagella? So we found out a different mechanism, and it's very similar to a organism called Microscopy xanthus. So Microscopy xanthus does not use flagella, but it uses type 4 pili. And basically what it does is, like a snake, it will go to one side. And then when it wants to change its direction, its leading side will become its lagging side. So what is the relationship between Salmonella and Microscopy xanthus? So these are filamentous cells. And there was some filamentous cells can also become motile. And this is an example of that. So this is actually Salmonella Newport. And see, it goes up to a corner. It reaches the corner and it has to change direction. And so it just goes off in the opposite direction. So it is quite interesting to see that Salmonella Newport can actually have motility similar to Microscopy xanthus. So again, another phenotype that has never been seen before, we can see that Salmonella is able to do it. You can see most of the cells are non-motile except a few. So this is another method. This is another unique mechanism that we are seeing as a response to antimicrobial stress. So filaments are a survival mechanism, and there are serotype-based variation in stress responses. It is important to understand these environmental triggers of filamentation because it is more common than we think is happening. So on the top panel, you can see a picture of Salmonella exposed to sunlight. And even after being exposed to sunlight, they can form filamentous cells. So filamentous cells might serve as a Trojan horse strategy, and this might help Salmonella survive. But so I would just like to introduce you to the people that I've worked with. So to the left is my advisor, Dr. Shirley McAuliffe, during my postdoc. And then here are my collaborators, Dumitru Makarison, Dr. Abhinav Mishra, and Dr. Kevin Missolval, so Dr. Abhinav Mishra and Dr. Missolval are working with me at the University of Georgia in Griffith. And Dr. Dumitru Makarison is at the FDA. All right, so again, this is my email address. If you have any questions, please feel free to contact me. And this is a heat map of my lab. So these are my two students, Kalen, and the other student is Aiki. Aiki is from Nigeria and Kalen is from Michigan. So thank you very much. And if we have time, I'll be happy to take any questions.
Yeah, thank you very much, sir. And participants, if you guys have any questions, you guys are welcome to ask. You can also post the questions in the chat box. And you are now allowed to unmute yourself so you can ask it if you have any queries. Okay, students, please feel comfortable to ask any questions if you have. You can also post it in the chat box.
Yeah, you don't have to only stick to Salmonella. You can ask me about
Yeah.
graduate education at University of Georgia if you want to. So I'll be happy to answer any question that you have.
Regarding your higher studies or anything related to microbiology or food safety and public health?
Yes, any question I'll be happy to answer. And yes, the University of Georgia has a great food science program. Our department head is Dr. Manpreet Singh. There are a lot of exciting food chemists, food microbiologists. There are people who focus specifically on bioinformatics. They develop pipelines for Salmonella detection, E. coli detection. Then there's me who studies phenotypic variation. There are people who study foodborne parasites. So it's a lot of expertise. Kevin, one of my collaborators, Kevin Misolval, he works with nanotechnology. So we are able to develop like nano sprays or nano powders to also fight foodborne pathogens. So yeah, it's not only microbiology, there are other areas of interest as well here.
Sir, there is one question from a participant. They want to
Yes.
know if there are any novel technologies to overcome the biofilm formation.
Yes, so that's a good question. So biofilms are really difficult, very interesting to study, but very difficult to eradicate, right? Yes, sorry. Yeah, so I think there's been some work on this compound called ursotic acid, which is basically in tulsi. So this compound from tulsi has actually shown to reduce biofilm formation in both E. coli and Salmonella. There are some fatty acids and there are some quorum sensing compounds that can also reduce biofilm formation. So these are some of the novel technologies that I've seen. But again, it's really difficult to eradicate a biofilm. So it's more fun to study them.
So I hope this has answered your question. Thank you for asking. Okay, so I request Mr. Shiprasad to conclude the session.
All right. Thank you very much for your time.
Thank you, sir, for coming.
Thank you, sir. Thank you very much for your talk on the topic filament formation by Salmonella is stress response strategy. The presentation was indeed interesting and informative, and I'm sure all the participants have been benefited with the information. Thank you very much, sir, for sparing your time in your busy schedule and then joining us for this conference. Thank you very much.
Thank you, sir, again. And we'll be starting with the next speaker now. Sir, there is one more question from a student. How about the masters in University of Georgia, or students of food technology? Govindraj, sir. Okay, we will start with the next speaker, next presentation. We will be just starting in a 2 minutes now. Okay, so we will be starting with the next presentation. I please request Mr. Shivprasad to give an introduction to the speaker.
Thank you, Kumkum. It is indeed a pleasure University College of Dublin, Ireland. Professor Tiwari worked as a scientist at Indian Institute of Food Processing Technology, India, before joining as a senior lecturer at Manchester Metropolitan University. He is currently working as a principal research officer at TGAS, Ireland. He is also an associate professor at University College Dublin, School of Biosystem and Food Process Engineering. Dr. Tiwari is a pioneer and an internationally acclaimed researcher working in the area of food processing technology with a special focus on novel technologies. His current research relates to the investigation of green and sustainable technological solutions for food industry. He is currently leading a team of researchers comprising 8 postdoctoral researchers and 16 PhD scholars at TGAS who are working in the area of novel food processing technology. Dr. Tiwari is an outstanding researcher as well. He has more than 180 peer-reviewed journals article, 108 international conference paper, 100 book chapters, and 1 patent to his credit. He was also named as one of the most cited researcher of 2018 by Web of Sciences. Dr. Tiwari is currently a fellow of Royal Society of Chemistry and Institute of Food Science and Technology. He is currently the editor-in-chief of Journal of Food Processing and Preservation. He is the guest managing editor of several other journals such as Food Research International and Innovative Food Science and Emerging Technology. He is also a member of the editorial board of Food Engineering Reviews and Ultrasonics and Solar Chemistry. Sir, it is a great honor to have you as one of the keynote speakers for this conference. I shall now request you to kindly start with your presentation. Thank you very much.
Thank you, Shiv Prasad, and thank you for the introduction. So today I'm going to talk about novel sustainable processing technologies for ensuring food safety and security. And one of the things is I just miscalculated the time because I was supposed to travel, and it's morning 4 o'clock in Dublin. That's fine for me. And so this is I've already been introduced, and my key area is all related to novel technologies, whether it's processing or preservation. And I'm going to go through this. And I work for Chagas, that's Irish Agriculture and Food Development Authority, and we deal with almost every aspect of agri-food system, ranging from agriculture, grassland, crop, environment, food, rural economy and development. And as you can see, this is Irish map. And you can see we are located almost in every county, and we have a number of different centers. And there are mainly 2 food centers. One is in Dublin, that's the capital city of the country, and other one is Cork, which is 250 kilometers away. So as you can see, our main focus is mainly related to sustainable agri-food system, which is a current challenge. And I lead a research group, as Shiv Prasad already mentioned, and we deal mainly with non-thermal technologies, novel extraction technologies, and novel ingredient. And you name it, we have it. That means we have almost all the technologies at lab scale as well as at pilot scale. And our key focus is mainly associated with how we can extend the shelf life of food product without impacting food quality, maintaining the nutritional attributes, adding value, and how we can make utilize waste, both evitable and inevitable waste. Now, before going actually into the topic of sustainable food processing technologies for food safety and security, I would like to put some emphasis on what do we really mean by sustainability, because this is what I personally feel, this is a bit of use and abuse of word sustainability. Because if you look into the graph, if you plot sustainability across time, suppose we are running a system, take example of food processing. Suppose we use for previous speaker was talking about on different aspect of a very interesting presentation. Suppose we are using chlorine for decontamination or for washing of fresh produce. And all of a sudden FSSAI in India or EFSA or FDA they restrict the use of chlorine for fresh produce. What happened? That is a disturbance. That means systems takes a hit. What do we do? Do we have an alternative? How do we make product safe for consumer? So system has taken a hit, and then system absorbs that pressure, that we get an alternative or we come up with some other solution to replace chlorine for fresh produce. And then we try to recover and then adapt to a new change. And when we adapt to a new change, the system becomes either performs exactly the same the way it was before, then it becomes resilient, or it doesn't perform the way it was previously. So it becomes anti-resilient. So, and these disturbances can be economic, social, environmental. And if we take all these functionality together, then we can call it a system as sustainable or unsustainable. So one given process cannot be considered as sustainable over other process, but it's amalgamation of economic, social, environmental. This also perfectly fits with the current situation of COVID-19. For instance, this would have been a physical conference. Now we have taken a different approach, going on electronic conference. So resilience, capacity over time in the face of disturbance, whereas capacity to preserve in the long run will be a sustainable. So I use terms sustainability and resilience interchangeably, but keeping the trend, leaving as a sustainability. So what do we mean by sustainable food production? What is the purpose to achieve that? To build a better future, that's a long run. Mission, creating sustainable value chains by providing food that satisfies the consumer need. And what are the values? Food safety, quality, reduction in natural resources, recycling and reuse. That is 3R. Reduce, recycle and reuse. So in sustainability, we mean environmental, economic and social. And together, we focus on green economy. So if we take a food cycle, ranging from production, processing, consumption, and recycling, we'll try to focus mainly on new technologies in the next couple of slides on production and processing, ensuring food safety and security, not going to focus on consumption and recycling. And one thing has been emphasized in the European level, it's if you really want to see sustainable growth, that we have to embrace new technologies. We say new technologies, that means to have a disruptive technologies. For instance, the use of chlorine is continuing for so long. We need to find alternatives to chlorine because of negative aspect of chlorine, environmental issues, health issues, and resistance against those chemicals. So in a nutshell, why do we really need new technologies? What's wrong with the current processing attributes? First and foremost is regulation. As you all are aware, there are a lot of documents has been issued worldwide about restricting the usage of pesticides in food systems, in agriculture, restricting use of preservative in food. So we need to find an alternative to that. So that's one of the reasons why we need to look for new technologies. Second important point here is surface cleaning and disinfection. In most of the cases, thermal processing is still in use and is a very good way of making food safe. However, what about fresh tomatoes, fresh apples, fresh lettuce or salads? What we should use to make it clean and disinfected? Because E. coli, Salmonella, as what our previous speaker was mentioning, is a greater concern. How we ensure food safety while extending the shelf life of product? Whether thermal processing can be replaced with the new technologies, whether we can retain nutrient and sensory aspect. Demand from consumers as well as processors to have energy efficient technologies. Apart from that, there are technological advances in various other fields, maybe non-food areas, and food processors would like to see those advances in food processing chain. In order, they can reduce the environmental impact of their food processing or agri-food chain. So hence, these are a couple of points where I feel why we need novel processing technologies. Now, what really mean by novel technologies? Because I will quote some of the examples of the technologies. In coming slides, maybe high pressure, pulse electric field, the range of different techniques, which can be classified as electromagnetic, biotechnological, mechanical, or thermal. Novel thermal, for example, dielectric heating, radio frequency heating. Novel non-thermal, how do we classify novel thermal and novel non-thermal? Is non-thermal, we are not applying direct heat. Heat is generated indirectly. For instance, in high pressure, it's we achieve high temperature at ambient condition, we are able to achieve microbial inactivation. So combination of these is the best to obtain a smart solution for it, to ensure food safety, improved shelf life, retain nutrients, improve processes, and which can be considered as environment friendly. So it will be very difficult to cover almost all the technologies, all the novel technologies which can ensure food safety in such a short time. But I'll cover basic about most of the non-thermal technologies and with specific examples. One of the most important question which comes up, like what technology we should use for A, B, or C product. So in that case, the most important aspect for selecting a technology is to understand their key mechanism and what we are aiming to achieve. For instance, if it's high pressure, the key mechanism is associated with pressure. Pulse electric field is electroporation, formation of pore due to electric current. Ultrasonics, it's pore formation due to sound pressures. Cold plasma, UV light, and ozone, all is related to oxidation of cellular matrices or oxidation of key compound. Thermal is all related to thermal degradation, that's heat. So if you look into this plus and minus signs, it just shows how different technologies impact the cells. So if we have a target microorganisms, and on what basis we can inactivate. So either having the combination of technologies or using a technology alone can cause microbial inactivation. So key mechanisms associated with vegetative cells are damages to cellular membranes, inactivation of key enzymes which modulates the growth of cells, oxidative damage to cell membranes, and damage to DNA or nucleic acid. And when it comes to spores, maybe Clostridium botulinum or other source or spores, is mainly damage to the spore coats or chemical modification in core of the spore or cortex. So this is what table summarizes the key mechanism associated with the range of different non-thermal technologies. So it won't be justified if we do not look into the chemical aspect of these technologies, because often we know a lot about microbial safety using these technologies. A lot has been published, a lot has been investigated. But in majority of the cases, whatever novel technology we take, they have very limited effect on macronutrients. Sorry, very limited effect on micronutrients like vitamins, bioactives, and minerals, whereas they have profound effect on protein and lipids. For instance, high pressure causes oxidation, denaturation, aggregation of protein molecules or proteolysis. Lipid, mainly oxidation, lipolysis. As you can see, plus and minus, that clearly shows what is caused by what technology. For instance, if we use meat for high pressure processing, we see a high amount of oxidation because for every 100 megapascals increase in pressure, there's 3-degree increase in temperature because of moisture, or 9-degree increase because of fat content. So pressure conditions and impact on the quality of the product largely depends upon the lipid and protein content of the compounds. Same way for ultrasonics and same way for plasma. So each of these effect are mainly because of oxidation, proteolysis, or lipolysis. So that's that shows what will have significant effect or moderate effect on different technologies. Now, how do we ensure food safety and security? The key approach here is to reduce the wastage. How we can reduce the wastage in food? By increasing its shelf life, and to make process more sustainable. That means we have to limit the consumption of energy and water and to reduce the waste. So this is what we are going to focus on, and I'll quote three examples of 3 case studies. mainly related to high pressure processing, because a lot of commercial application has seen are there exist at the moment for high pressure processed food product. Other one is ultrasonics and plasma technologies. So with this first one, high pressure, in this is relatively very simple technique where you have a product which is packed, and then whether in a batch mode or semi-continuous mode, it is subjected to significantly higher pressure. It can be up to 800, up to 1000 megapascals. However, industrial setups, most of the industrial processes do not want to increase pressure more than 600 megapascals. Only reason is because of strength of material and making high pressure process industrially relevant. So here, this is one of the most simple thing. It's just most of us, we use pressure cookers at home for cooking food. It's very similar to that, where you can see the industrial machine over here, where we load packed product, something like that, and load into the high pressure machine. Now, we carried out commercial trials, and we carried out a commercial validation process for Village Dairy on milk with HPP tolling, because every company cannot afford to purchase high pressure equipment. So in Ireland, there is a company funded by Enterprise Ireland. That's one of the departments, Department of Enterprise funded scheme, where they have a big high pressure unit of 200 liters, and processors can bring in their product, get it pressurized, and can go for commercial market. So in this particular study, we were trying to compare high pressure processed versus thermally processed milk. So here, milk, raw milk, compared to pasteurized and high pressure milk. So we looked into the shelf life over 28 period time on the condition where we were able to achieve 5 log reductions in pathogenic microorganisms. And what was found? We found over 5 log reductions for pathogenic microorganisms, more than 10^6, then more than 6 logs reduction in total viable count, lactobacillus and pseudomonas species for 21 days in high pressure processed milk. And quality, there was no significant change in color, and high pressure yielded significantly smaller fat globules. That means it also provides homogenization effect. For instance, if you look into thermal processing of milk product, we heat a milk and then it goes through homogenizer to make milk consistent. Otherwise, it will make a cream layer, or it will have larger fat globules, a separation of fat. Something like when we boil a milk, we get fat separated. So in the case of high pressure processing, we are not only able to reduce the microbial load, but we are also able to get high pressure, we are also able to get homogenization effect. So this is another example where we were investigating use of high pressure processing for fish application. So here again, fish is one of the commodity which really gets spoiled rapidly. So the main purpose of this particular project, which was a collaboration with the Polish institute, that is National Marine Research Institute, SINTEF, that's a private research company in Norway, and University of Perugia, that Italy, and Norwegian Technical University, where we adopted high pressure for fish. And interestingly, we did observe reduction in viable counts based on different conditions, 100 megapascals, 300, 500 for 2 minutes or 5 minutes. And as you can see, all these treatment conditions are within 600 megapascals. But if you look into the literature, you'll find it says if you go for 800 megapascals, you get faster inactivation. But that is good for academic exercise, but for practical application, it has to be less than 600 megapascals. Now, so when we looked into different aspects of viable counts or different hydrogen sulfide producing bacteria, or TBAS, that is lipid oxidation, we do find some changes in oxidation. However, one of the effect is the fish, which if you look at these images, the normal fish is having slightly reddish color, whereas when we pressurize the fish, it becomes slightly whitish in color, that is, it's having bit uniform color. So the advantage here was if we combine both high pressure and smoking of fish, we get very uniform color fish compared to red streaks what you find only on smoked fish. And this is only because the red color becomes darker, whereas smoked plus high pressure improves the product quality. So when it comes to color values, we do find it much more consumer appealing. We have less formation of certain compounds which can be considered toxic and extend the shelf life of fish product. So here, high pressure can cause microbial activation, but can have changes in color of the product. But that can be taken advantage if we know we use it for specific applications. So now this is very interesting project. It was industry-led Horizon 2020 project, where we were trying to validate high pressure processing for ready meals. And this is a short video which will run here, and I'll discuss with the results.
High quality processed food is rapidly growing. To satisfy these needs, the food industry has to innovate. The Hipster project is developing the latest state-of-the-art food processing solution. HPT, hydrostatic pressure combined with temperature, will enhance the quality, shelf life, and safety of food products, whilst reducing their global footprint. We're currently designing new equipment to introduce HPT to the global market. And this is how it will work. The food product, sealed in flexible and water-resistant packaging, is first inserted in a container and then preheated. In the next step, the preheated food is subjected to an enormously high pressure of up to 6,000 bar. This pressurization lasts just a few minutes and results in an instantaneous increase of the temperature up to 120 degrees, enough to destroy the most resistant microorganisms and enzymes. The short pressure and temperature treatment ensures that the quality of the product is hardly affected. Through extensive research, we will be able to...
Okay, now this shows high pressure and temperature in combination. The reason why combining high pressure and temperature together, if you recall when I was mentioning about smart combination, was mainly related to spores. Two spores, that is Bacillus amyloliqueficans and Clostridium sporogenesis. These two are very, very resistant to temperature. As you can see from this graph alone, even at 110 and 115 degrees Celsius, we see up to 60 or 80 minutes, we are not able to get more than 5 log reduction. Whereas when this is adopted along with the high pressure, we are able to achieve the same reduction within less than 5 minutes. So that means high pressure and heat can inactivate spores, whereas spores cannot be inactivated with high pressure. So combination of high pressure and heat, that means high pressure opens up the spores and heat inactivates the spores. So that's the combination for both Clostridium sporogenesis and Bacillus amyloliqueficans. So this clearly shows within 3 minutes we are able to get more than 7 log reduction in these very resistant, thermally resistant spores, and that was observed for vegetable soup, steamed sole, veal soup, and green pea ham. So this is what it shows, how we can combine heat plus pressure together to make a machine which is now commercially adopted. So that was the example of high pressure. We looked into the liquid food, semi-solid food, but one of the disadvantages about high pressure processing is we cannot use it for solid food because it needs a moisture, and it cannot be used for carbonated beverages, and high pressure cannot be used for beverages or product which does not contain a moisture. Now, second interesting technology, what I was planning to discuss was ultrasonics, which I've been working for last so many years because I feel this technique is very intriguing. The way it can be used for different applications based on mechanical and chemical and biochemical effect. And if you look into ultrasonics, anything above 20 kilohertz frequency will be considered for processing applications, and anything above 2 megahertz or above 5 megahertz goes for diagnostic ultrasonics, which you use for medical purposes and also for developing various types of sensors. So majority of the processing applications are between 20 kilohertz frequency up to 1 megahertz frequency. So that's the range which can of cause crystallization, degassing, extraction, encapsulation, drying, chemical biochemical effect like bactericidal effluent treatment, modification of growth of living cells or inactivation of cells, and different applications depending upon the frequency and the power we use. Now, this particular technique is different from contact type ultrasound. Normally, ultrasonic bath you would have seen in most of the laboratories where you need to have a medium, that's water or oil, to deliver cavitation to the product. Whereas airborne acoustic is a technique where there is no media required. It's only non-contact type where air can be used for decontamination purposes or for process intensification. So airborne acoustic technique is something can be used for defoaming application and is being used in commercial space. It is also used for reducing foaming loss. For instance, if you look into the beverage processing line, where you have filling of carbonated beverages, maybe Coca-Cola or any of those beverages, as you can see at the corner where my food is coming up, you can see the formation of foam, and there is up to 1 to 2% losses of the product. What if there's airborne acoustics, that is separation of sound pressures, it will avoid such losses. So these are some of the applications which are currently ongoing. And what we were trying to investigate is how we can use non-contact ultrasonics for microbial inactivation. For instance, if you take a starch or if you take a black pepper, so these are the products which are readily used for as an ingredient for topping up on onto the products for adding a black pepper. But what technology do we have to cause microbial inactivation in dried ingredients? For instance, starch. Starch extraction process never goes more than 40 degrees Celsius. And we get dried starch and we use it for a number of soap mixes. Same way for black pepper and other dried ingredients, spices, pepper, they do have chances of getting microbial inactivation. So we were investigating airborne acoustic to achieve such type of microbial reduction. And we employed it for black pepper and for tapioca starch. This is just a model food. And what we observed with non-contact ultrasonics, we are able to achieve more than 1 to 2 log reduction for black pepper, same thing for spores. But time is way too high. But one good thing is airborne acoustic technology is also used for drying of food products. So if we combine airborne acoustics for drying of such ingredients, for example, for black pepper, when it is harvested, is having normally moisture of 18 to 19%, it is hot air dried, which may not be suitable for thermal causing inactivation in microorganisms. But if airborne acoustic is used, we can achieve both drying as well as microbial inactivation. And even such a long treatment time up to 60 to 90 minutes, we do not see any significant changes in phenolic content of black pepper or rheological properties or viscosity profile of starch. Now, this image where what you see ROS, that's is basically in order to reduce the time of treatment, we try to introduce plasma technologies along with airborne acoustics, because plasma can cause inactivation. I have some of the examples in coming slides. However, how to make sure that the plasma gas falls onto the surface, because it's very difficult to direct the gas. So if we have airborne acoustic and plasma gases together, we can reduce this inactivation time to much shorter length. So I'll quote, I have some examples for that as well. For instance, log reduction for spores contaminated samples, we were able to achieve very less amount of spores inactivation, whereas when it comes to vegetative cells, we were able to get significant amount of microbial reduction. And as you can see, this is Bacillus subtilis cells, and it just shows disintegration of cells because of acoustic pressures. Now, there are significant mechanisms has been known for acoustics. That's mainly for microjet, microstreaming, as well as acoustic streaming. And most of the inactivation mechanisms or action of ultrasound are all related to cavitation bubbles. Now, I'm not going into great deal of it, as I can see I might run out of time. So main mechanisms associated with ultrasound are all related to cavitation, and this is mainly applicable to contact type ultrasound. And if you remember, when I mentioned ultrasound can be used for increasing the microbial growth of cells, you can look at this image. This is Lactobacillus sake cells. We can cause a controlled pitting. In this case, we can control the growth of cell, and cell death only occurs when we have a sufficient amount of time or exposure time which disintegrates the cellular membrane. And in these cases, in B and C level, we are able to increase the cell growth because of higher mass transfer. That means increase efficiency of taking a substrate and excreting out the compounds, and also disintegrating the cells. That means improving the cell surface area. So that's the way ultrasound seems to be a very interesting technique. Now, as I mentioned about ultrasound using with the plasma, and plasma was briefly mentioned by our previous speaker, and in simple term, plasma is basically air which is charged to a higher voltage, leads to the formation of various reactive species. And these reactive species depends upon the gas composition. It can form reactive oxygen species or reactive nitrogen species. When we have oxygen, it forms only ozone, but we have air with nitrogen, carbon dioxide, it forms a range of different reactive species. And that particular reactive species causes microbial inactivation. For example, if you take a food product, in this case is a meat, packed in a specific air, put into high voltage discharge, it causes reactive species formed, causes microbial inactivation, and these are such a short reactive species, they go into their stable state quite quickly. So where we can use this cold plasma, we can use it for a range of different pre-harvest, post-harvest, as well as processing applications. Fresh produced grains, fish, dry food, meat as a replacement for pesticides, and food processing industry. So this is one of the most versatile upcoming technology, and however, regulation will have some impact on it. So these are some of the plasma techniques what we have at Chagas, and we have been constantly developing in collaboration with the Electrical and Electronics Engineering Department of Dublin City University. And how we can use it in food application. For instance, packed in air, and we get extension of shelf life. And another way is plasma jet, getting a plasma activated water, getting these reactive species in the water, and replacing chlorine by using different type of product immersed in the plasma activated water. And various reactions which takes place, that is again ozone generation, nitrous oxide, or formation of various other reactive species. So similar applications are also for biofilm. So again, various reactive species causes oxidation of organic matter. So that's some of the way how we can use plasma. Now this just shows how plasma activated water causes microbial load. So as you can see, this is a jet which is injecting plasma into the water, and you can see how much log reduction we can get it for E. coli or Pseudomonas, and the cause of mechanism is again the oxidation of cellular membranes. So we investigated plasma activated water on range of different microorganisms relevant to food and other areas. Now, if you look carefully, watch this particular video, where you see the plasma air. If a food is kept in a plasma-rich air, we do not see a microbial growth. That means if we are not using plasma directly onto the food surfaces, we can always use it as a processing aid. For instance, if you look into this particular poultry house, one of the interesting study which was carried out with our industry client in Europe, when air is stale, like if you visit any of the animal house or livestock, we see very foul smell coming out. And because of that, what happens, chicken, they eat and they sit at one place. So they develop more fat, less of protein. And when we use plasma in such environment, it purifies the air by oxidizing all those gases, all those organic matter, which causes foul odor. Foul odor. In that case, chicken moves a lot and they develop more protein than fat. So these are just some of the hypothetical things how we can use it for food grain and seed storage, because it can inactivate microorganisms, bacteria, pesticides, and can replace pesticides. And this is one of the prototype which was developed for tomatoes, and where we find extension in shelf life. Moving forward, and how we use both airborne acoustics and plasma activated water for biofilm, because this is one of the most important aspect of biofilm. I know we have another speaker who will go in detail with the biofilms. So I'll just quickly present some of our results. As you can see here, when we used airborne acoustics and plasma, we get significant reduction in viable cell counts and significant reduction in biofilm biomass. So airborne acoustics, that's non-contact again, and plasma together can inactivate, can remove biofilms, both inside a tube and on the surfaces. So again, mechanism of action is all related to the chemistry. I'm not going into details of it. So I love this slide because it makes me look like I'm a big chemist, but that's not the case. We do get help from our proper chemists to interpret these data. So main mechanism associated with that is dissociation of gases into various reactive species, and these reactive species is having huge amount of microbial load. And these reactive species can be controlled by controlling the type of gas we use it in plasma systems. Now, after going through three technologies, high pressure, ultrasonics, and plasma, how do we use these technologies? Because if you look into the literature, majority of the studies reported are on model solutions where we can easily achieve to 5 log reduction. When it comes to liquid food, still we are able to achieve 5 log. But when it comes to solid food, whether it's fresh produce, you have fish, meat, or cheese, or any other dry food, we do not get more than 2 log reduction. So if you look into US FDA, there is a law, if a technology is able to achieve 5 log reduction in liquid food, that should be considered as non-thermal technology. But in solid food, getting even half a log reduction is a challenge. So if you look into the chlorine application, most of places we get only 1 log reduction. 1 log itself is 90% reduction. Most of us are using disinfectant. It says 99.9% reduction in microbes. That is basically a 3 log. So how do we use these new technologies? First and foremost is food safety, microbial inactivation. If it is able to achieve, then how do we integrate into the processing chain? And then to check into the processing efficiency. If it is no, and then try to find a suitable combination. Something is missing here. Suitable combination. And then if you are able to achieve that, then we go for commercial validation. So the biggest challenge at the moment is we all are into basic research. That means make a small system, prove that it works. The biggest challenge lies, how do we make technology transition to commercial system? So this has been a quite big challenge, and most of the projects which are funded at our place are mainly at different technology readiness level. So industry wants a TRL 9, that is system test, launch and operation, whereas basic research is easy to establish and at lab cases. So far, whatever I talked about was all about goodies of different technologies, but what is not known of novel technologies is the toxicological aspect. That means what processing induced chemicals it may form, or is it producing certain other chemicals which may cause which may be of public health concern. For instance, when we use UV, ultraviolet light, we may get good inactivation rate, but if we use it on a meat, will it oxidize the cholesterol to dehydroxycholesterol, which may be a toxic? Or if it is causing a protein oxidation, does it form certain carbonyls which may have negative health effect? So our now more focus has shifted toward toxicological aspect of new technologies, and is there any impact of new technologies on bioavailability and bioaccessibility of nutrients? Because microbial safety, we all know about it, it's not been reported, shelf life and quality as well as nutritional attributes. So this one is one of the most important aspects which requires further investigation. Apart from that, in order to ensure the sustainability of these technologies, it is also important to characterize these technologies in terms of environment, natural resources, economic prosperity, human health, and social well-being. So together of last two ones, assessing the sustainability aspect of these new technologies, then only we'll be able to make sure that yes, these technologies can really have a positive impact on agri-food systems. So to conclude, novel technology solutions are available for a range of agri-food systems, and these technologies and practices that support balance between production efficiency, food quality, and safety is the paramount. And not a single technology, not a single technology can solve all the food problems, but it is all the combination of different approaches, thermal with high pressure or ultrasound with pressure, or combination of different technologies can provide a package where we can ensure sustainability and can ensure food safety of food product. And whatever was presented here was part of our research work, ongoing research work. It's not that all I do it. Of course, I do take a credit of all this. This is my team, which is currently working on various aspects of new technologies. And we have generous funding from various funding agencies, including industry. So with this, I finish my presentation and happy to take questions.
Okay. So there is one question in the chat box from a participant. Sir, you just mentioned that novel technologies affect the macronutrients and thermal technologies affect micronutrients. So the participant wants to know, is there any technique that will help in retaining both micronutrient and macronutrient along with increasing the shelf life?
Okay. So when we process anything, right? When we process anything, there will be a impact on the nutrients, whether the heat or any other technology. The best case scenario is when we try to process a food product, there will be some negative effect on both macro and micronutrients, and the selection of the conditions will be largely depends upon on the best case scenario, where we are able to achieve food safety objectives and have a minimal impact on quality. For instance, if you're trying to replace, take for example, orange juice, if we heat to 92 degrees Celsius as a thermal processing, we get up to 1 to 2% reduction in ascorbic acid content of it. But over the storage, it causes changes in further reduction. Whereas when we use ultrasonics or high pressure, we do not get any changes in ascorbic acid content. In fact, studies report there is slight increase in ascorbic acid content. It's just not. It's just because amount of extractable ascorbic acid has increased. So it largely depends upon the technology as well as onto the product what we are trying to process. Similarly, when we use apple cider for ozone, we get microbial safety with minimal impact on nutrients. But if we use the same ozone for strawberry juice or for orange juice, we see significant changes in anthocyanins and other phenolics. So there are technologies, but it has to be, as I mentioned about selection and integration into the system, is how best we can reduce the impact on the nutrients. So it will be very difficult to say which technology is good. That will largely depend upon type of the product we want to process.
Okay, so I hope that has answered your question. So there is one more question. Are there any novel technologies for preserving fermented foods?
Yeah, it's actually, as far as fermented food is concerned, if you look into the type of microbial contamination you will receive, we will get in fermented food is all related to molds, mold growth, and some bit of bacterial contamination also you get if it is fermented meat or if it is fermented vegetables. So there are two different things. If it's fermented meat, there are also issues of lactobacillus and some of those spoilage microorganisms. And that high pressure itself can do that particular job, because most of those fermented products are high moisture product. And high pressure will be much better approach to deal with such type of wet fermented product, whether it's gherkins or it's fermented sausages or so. When it is fermented semi-dried product, then again, high pressure will be the best one, or to have a in-pack plasma treatment can do the job.
Okay, thank you, sir. There is one more. Why is novel technology not used for solid foods?
Well, it is novel technology is used for solid food. For example, ozone. I quoted example of plasma for solid food such as fresh produce. That's a solid food. For grains, it can be used. But majority of novel technology, for example, contact ultrasonics, pulse electric field, high pressure processing, all these technologies are suitable only for liquid food, because the type of delivery they pose. So that's one of the reasons. For instance, we cannot use high pressure processing for preserving grains. And there's no requirement of using high pressure for grains, because grains itself is a self-stable product. Whereas for that purpose, we have to use ozone or plasma gases in order to get rid of insects in it, to avoid usage of pesticide, or to inactivate molds, which may cause mycotoxins. And when it comes to another solid food, we can call it a solid food as a bread. For that, UV treatment, because most of the bread is affected on the surfaces side of it. High pressure cannot be used for that purpose because it will damage the product integrity. So these are some of the challenges. Even we can use high pressure for apples, but it will give a cooked flavor, and there's no requirement for using high pressure for apples. Instead, we can use surface cleaning agents, for instance, ozone containing water, or using plasma activated water or plasma gases.
Yes, sir.
Yes, sir. Yes, sir.
Yeah.
Hello. Am I audible?
Yeah. Yeah, I can see one of the question, how good is pulse light technology for fresh cut vegetables? Yeah. Well, obviously pulse light technology, yeah, that means using it. If you're talking about white light, yes, it can be used, because some of the studies are also with the blue light, red light, UV light that can be used. But when it comes to pulse mode, because we are working on LED light on the pulse mode, and none of the physicists, they agree using pulse mode of light will have any effect. But most of the food scientists, they do believe that when we put light in a pulse mode, we get better efficacy. But on the physics side, they do not see any relevance of using light in a pulse mode, because it is all amount of energy which is released by a light is absorbed by a microorganism that causes microbial inactivation. And especially when it comes to whole white light, That means talking about full spectrum of light between 380 to 780 nanometer or 800 nanometer. That's all the energy which can be used for fresh produce. Whereas personally, when we started working on UV LED light system, we prefer to use a specific wavelength LED light because 253, 254 nanometer is good enough to cause microbial inactivation. Whereas if you expose to whole bandwidth of light, it will cause the oxidation of the product. So it is good to use light for inactivation. That's whether it's UV light or white light or blue light. And that's all related to energy absorbed by the product. What if we select, if your aim is only to get the microbial inactivation on the surface, we'll pick a specific band of wavelength rather than having a negative effect of whole range of bandwidth on quality of the product. Because certain light technologies are also used for horticulture applications for growing off-season crops. Even when it comes to potatoes, we need extended light zone to get potato seeds.
Okay. Thank you, sir. There is one more question.
Mhm.
Is it safe to use the smoking preservation technique along with high pressure processing, since smoking is known as carcinogenic?
Yeah, smoking is carcinogenic, but at the same time, smoke products, like we all know, whether we use, I'm just going on, somebody was looking for my contact details, so I'll just go on to the first slide. So it's smoking produces carcinogenic product, we all know that. But at the same time, smoke product, whether it's even if you eat a lot of tandoori items, they all are made in basically a smoke product. So flavor and fragrances are also important. So in that case, when we use high pressure and smoking, we did find less effect on those carcinogens. So I could not present all the data there. So some of those smoked element also, whatever is smoking produce certain aldehydic and ketonic group, and it causes, oh sorry, it's not, yeah. So it causes certain compounds which are also antimicrobial. So when we use high pressure followed by smoking or smoking followed by high pressure, we get improved shelf life because of those certain antimicrobial compound formed due to smoking increases microbial inactivation. Yes, certainly it forms carcinogens, and moreover, if you use thermal processing, it forms Maillard reaction. One of our research collaborator in Denmark is working on cancer causing agent in Maillard reaction. So there is no never end. There will be always compound formed which may have negative impact on health.
Okay. Sir, would you like to take a few more questions?
Yeah, go on. Yeah.
Yeah, okay.
Yes, sir. Good morning. So there is a question like they want to know, like, you have talked about the application of plasma technology on poultry farms to clean the air, deodorize the air. So is there any particular health effect of those chickens, poultry chickens, they are getting exposed to those plasma, and is there any health effect of plasma technology or exposure to plasma on human health also?
Okay. Now, when it comes to plasma, if you look into the literature, plasma is also used for medical applications. So plasma for surface treatment, because yes, these reactive species are not healthy for humans, of course, but plasma is also used for cancer treatment. We have two of our PhDs, they are working on usage of plasma for treatment of cancer cells, mainly on a brain tumor. And plasma for medical applications is used. For instance, argon gas is good for wound healing. You can look into the literature. I cannot put those examples over here. And if you are using plasma in food processing environment, and human exposure to that level will be toxic to them. However, when plasma is used for food product, it forms a reactive species for just for millisecond or microsecond, and which quickly goes into the stable conditions. So there are occupational health and safety, and moreover, when plasma is generated, it is generated with very high voltage in tune of 120 kilovolt to 90 kilovolt, and being near to that one is not advisable. So that's certainly that's having health and safety concerns, but it's also having application for cancer, but it is also having application for cancer research. It is also having application on wound healing, and we do not expose ourselves to very high concentration of those reactive species. And to be honest, that is applicable to almost all the technologies. For instance, pulse electric field, here we are talking about up to 12 kilovolt per centimeter, or per centimeter of a gap. So 12 kilovolt with higher current is really dangerous. So we have to be always cautious of any of the technology when we use.
Sir, there is one last doubt from our side. So it was really interesting to listen about novel processing technology. But can you tell the technical challenges that we face when it comes to the upscaling of this technology in Indian scenario?
So it's actually whether it's Indian scenario or anywhere, the problem is the same. First one is we have to prove it works. So we know high pressure has been under investigation since 1960s. Ultrasonics has been under investigation since then. The only issue was how do we, as you see the diagram what I had from the fish, from basic research to a higher level, is operational capabilities. How do we integrate into the existing processing line? So that's the one of the biggest challenge, because most of the food processing companies, they do have existing processes on. So rather than changing the whole process without knowing what will be the economic benefit, what will be the environmental impact, and without having real-time data, apart from that, to getting a regulatory approval. At the moment, only high pressure processing, you'll see some of the commercial applications. So it's not only Indian scenario, it's a global problem. First, to convince convince the industry to adopt that, and they will adopt it based on their real economic benefit of it. Number two is getting approval from regulatory agencies, and getting approval from regulatory agencies is important, and it is also difficult because unless and until we have all the aspect answered related to that. So there are significant challenges to that, and the only way to address it is to have a commercial validation trials. For example, for thermal processing, when we use it, or even if you're using a retort in your labs, there is Clostridium botulinum is index microorganisms. Some of these technologies, they do not have index microorganism. E. coli can be inactivated by high pressure within 1 minute in fruit juice, but it will not inactivate the spores. Whereas heat will inactivate E. coli, will also inactivate spores. So there are some challenges, validation rules. And apart from that, you also have to convince consumers about the technology. For instance, as you all are, or majority of you are engineering students, so when radiation is third mode of heat transfer, right? Conduction, convection, and radiation. And when we use light radiation, or when we use ultrasonic radiation, consumer perceive that one with the gamma radiation, and that's the way it has to be, you know. So education and making people aware that these technologies are safe, and providing relevant data for industry, and then making ways and means how these technologies can be integrated into the system. So I quoted an example of HPP tolling, where the government invested 2 million euros to have a high pressure tolling facility, where the smaller companies, they can come in, they use that facility and get their the product processed. So rather than having every company buying high pressure, so there again, you know, cost benefit analysis becomes different. From food scientist perspective is good. We get microbial activation, we get this improved shelf life, improved nutrient, but when it comes to cost benefit analysis, it doesn't solve the problem.
Thank you, sir. So there is one last question. How far is it safe consuming food that is treated with the radioisotopes, traces which contain radioisotopes?
Okay, that radioisotopes, that means basically gamma irradiated foods, yeah? That's what it means, yeah.
Yes, sir, like cesium-137 and cobalt-60, their irradiation process actually.
Or using radioisotope, yeah, that is cesium or cobalt. That is basically, oh yeah, just gave an example of ionizing radiation, right? That's gamma radiation or ionizing radiation. Well, products are definitely safe, and this is what is a perfect example. Radiation was one of the best technology which was applicable for a range of different food products. Gamma radiated onions reduces sprouting of decontamination of food surfaces or everything. And one of the biggest issue was why this technology did not see a commercial wider acceptance among consumers, because every time gamma radiation was related with what happens in Hiroshima and Nagasaki. That was the biggest concern, that the people were not willing to go for gamma radiated food product. But gamma radiated food products are still on sale. They come in different countries, even in Europe they do come in. Most of the spices, most of dried ingredients are gamma radiated. That is, and in India also, like I think bark works on various aspects of food, and it is definitely safe, and it does not leave any of those isotopes into the product. And I can see there's another question: is there any combination of LED, grow light, and pulse technology? Well, basically LED is light emitting diode which comes up with the specific wavelength, and this should not be confused with LED lights what we get on the streets. That's different. So when I mentioned about LED, that light emitting diode, that is basically for a specific wavelength. That means of 254, 354, 405, the specific wavelength LED light I was talking about. And light technologies is a whole bandwidth of white light, or a specific color band, whether it's a blue or blue light, or a red light, or the whole white light. Well, I haven't seen much of other light applications in food, but definitely there is a possibility of using LED light. Yeah.
Thank you, sir. And any participants have further more questions, you are free to contact. The contact details have been put up on the slide. So you can take the contact and you're welcome to ask any questions. Thank you, sir. And I request Mr. Shiprasad to please conclude this session.
Thank you, sir, for an interesting and very informative presentation. I'm sure all the participants have thoroughly understood the intervention of novel sustainable processing technology for ensuring food safety and security. Thank you again for taking your time off from your busy schedule and speaking to us. Thank you again, sir.
Thanks.
Thank you, sir. The next presentation will be starting soon. We'll be starting it in a few minutes. Okay, now we'll be starting with the next presentation. Our speaker is here. I please request Mr. Shiprasad to give a brief introduction about the speaker.
It is an honor to me to introduce our next speaker, who also happens to be my mentor, Dr. Neethu Kumbrataneja. She is a pioneer researcher in the field of food microbiology and biotechnology. Dr. Taneja obtained her PhD in biotechnology from All India Institute of Medical Sciences, New Delhi, India. Dr. Taneja worked as a postdoctoral researcher at Wake Forest University of Health Science, North Carolina, United States of America, before joining National Institute of Food Technology, Entrepreneurship and Management, where she is currently employed as an assistant professor at Department of Basics and Applied Sciences. Dr. Taneja has been working in the area of food microbiology, food safety, and nanobiotechnology with a special focus on novel intervention for controlling microbial biofilm and food diagnostics. She has more than 15 research articles, several international conference papers, book chapters, and 3 patents to her credit. She has been awarded with several grants by external funding agencies such as Department of Science and Technology, Government of India, Ministry of Food Processing Industry, Government of India, Ministry of Tribal Affairs, Madhya Pradesh Government, and Science and Engineering Research Board, Government of India. Ma'am, it is our privilege and honor to have you as one of the keynote speakers for today's conference. I kindly request you to take over the session. Thank you very much.
Thank you so much, Shiv, for a very elaborate introduction. And it was a great experience to have you as a research fellow at in my lab. And at the outset, I would like to, a very good morning to all of you, first of all. And I would like to thank the organizers for having invited me for this talk at a very nice conference which I have witnessed, and I've been listening to a lot of good lectures by eminent speakers here. And without wasting any further time, I would like to start my presentation. And I'll be talking on the microbial biofilms and its impact on food safety. Talking a little general and then proceeding to the work in my lab. And what are the mechanisms of biofilm formation, resistance, persistence, and how do we control it? I'm working at NIFTM, National Institute of Food Technology, Entrepreneurship and Management, as an assistant professor for the past 8 years. And my lab has been working in the area of microbial biofilms since the beginning of the research career here at NIFTM. This is a brief outline of my talk, and I'll be briefly starting with what is food safety and foodborne pathogens, their impact on food safety, how microbes grow in biofilms, how do they communicate with each other, and when do they decide actually to enter into the biofilm forming phase. What are the advantages to the bug and its relevance to everyday life as well as the food industry? What are the routine interventions, and one of the few works which my lab has been doing. In fact, in the morning, Dr. Govind had given a very preliminary overview of Salmonella growing under stress conditions. He spoke more on the filamentous Salmonella, and that also he was referring to the floating biofilms. So continuing, and maybe I'll be elaborating more on the biofilms and giving a more detailed overview of what they are, since a major group of this conference are students. So I'll try to make it more understandable to them, as well as sharing the research data. Thank you. So when we say food, we refer to a substance which provides us nutrition, which provides us energy, and which has to be very safe. We should not fall ill after consuming anything which is providing us energy, and we have to take in and day out. So the access, I would rather say access to safe, nutritious, and wholesome food is a fundamental human right. Okay, each one of us deserves to have a safe and nutritious food at very affordable prices. And food safety is the branch which is actually dedicated towards the conditions as well as the practices that preserve the quality of the food, as well as ensures that the food is safe to the consumer. It actually includes all the practices right from the stage of production at the farm level to to actually the growing it, harvesting it, packaging it, processing it, and making it available to the consumers to buy after packaging. It includes storage, distribution, preparation, consumption. So all the stages of food production till the time it reaches from the farm to the fork is a part of the food safety, right? And it is at each of these levels we have to ensure that there is no infiltration of any sort of chemical, physical, or biological agents to ensure that it is safe. And of course it is very relevant to the public health, because safe and unsafe food will lead to health issues at a wide scale level. So, foodborne diseases, as all of you know, are a global threat. They are a global health threat, and they are transmitted via contaminated and raw and processed foods. And they are majorly caused by a class of microorganisms such as Salmonella, Vibrio, E. coli, Listeria, etc. And if we see at this graph here, the global burden of mortality, uh, I don't know, I'm getting a lot of messages here of people entering. If the organizers, if I can avoid seeing these messages, it'll be better. Thank you. And so basically, the global mortality and the morbidity of the foodborne diseases has been really high. Over 600 million cases of the illnesses have been reported in the United States, according to the data of April 2020, and it led to over 420,000 deaths and an economical burden of more than 110 billion US dollars. And if we talk about India, we are really underestimating the burden of foodborne illnesses. We don't really have a very foolproof surveillance system yet. However, according to the data which is available from the FERG report, in the East Asian region, over 150 million illnesses due to food-borne diseases, and over 175,000 deaths, with a loss of 12 million daily time per year had been reported. And it's little old data, so it's actually very incorrect. You can multiply it by several fold times to actually know the actual levels. And what do they cause? The symptoms may be mild or they may be very severe. It usually causes diarrhea, abdominal cramps, nausea, fever, and vomiting. However, it may progress to really life-threatening diseases with kidney failure, or really even hemorrhage in any other part of the body. So it can be really fatal as well. If we do a reality check, what do the foodborne disease statistics tell us? These are the statistics of United States in a comparison with India. Okay, so if you see over the past couple of years, the trend has not reduced. There is a trend that Campylobacter, we can compare various foodborne disease causing agents, and the trend has not reduced. The incidence of foodborne outbreaks has largely remained unchanged in developed as well as developing countries like India. We see the illnesses as well as deaths caused for the past 10 years has not really changed despite of the interventions and the public health strategies the regulatory authorities have been taking. So why is it that despite of so many efforts being done, there is no so-called, really evident change? So that's a question to everyone, to all the professionals who are working in the area of food sciences, especially in food safety, that what should we do, and why is it that our such hard efforts are going in vain, and what are the reasons? So basically, what do we do in our regular food processing practices? We impose physical stresses on a food item, we add chemicals, or we may even do biological interventions. These are all the food preservation strategies so as to keep the food safe and ensure that the pathogen stays out of the food. So high temperature or low temperature treatment, pasteurization, we do dehydration, which is a really upcoming, I mean, it's already an established way of keeping the food safe. We do a modified atmospheric packaging, irradiation. There are high pressure processing and all the non-thermal processes which are being tried to ensure the food safety. A previous and very old and like usual practice has been addition of lots of chemical preservatives and antibiotics. Maybe even there are papers and reports where biological agents like bacteriophages and biopreservation has been a trend. So all these things that we are trying to impose are like stresses to the bug. The pathogen is basically forced to either succumb to the challenges that we are imposing, or it has to adapt to it. Okay, so what the microbe does, it senses. It has a very regular sensors. Okay, there are signal transduction systems in the microorganisms which sense the environmental cues and signals, and they basically either trigger and turn on and turn off the genes which are responsible for their adaptation to the changed conditions. Okay, so they either have a metabolic slowdown, they slow down their metabolism, they may sporulate, they may actually overexpress heat shock proteins, or they may basically have a blanket or a shield over themselves and form biofilms. So definitely bacteria sense and talk. Okay, and this is what biofilms happens in the biofilms. The bacteria communicate with each other just like as we all do, and they start producing certain molecules to basically ensure that this communication is a good one, and the phenomena is called the quorum sensing. So I would like to put in here that not all bacteria will communicate. If those bacteria which have certain genes for expression of quorum sensing, they will communicate and they will be the winners. Amongst the changed conditions of the food preservation strategies we are following, the microorganisms or the bacteria, they are basically the ones which are communicating, survive the stresses, and they propagate the infection. Okay, so here on the right hand side, I have just put in what are the basic adaptations microbes are having, and the biofilms are one of the major adaptations. So what happens in biofilms? There are bacteria which is sticking either to the host surfaces within our body, or you may say that it is a food surface. The bacteria are in a kind of a flux. They stick and then they settle down, or they are motile. It varies. When the environment changes, when there is a kind of a sense, so this will be a kind of a thing which will be a to and fro kind of a thing. Microorganisms will keep on, sometimes it sits, sometimes it stands, moves around and stuff like that. So when there is a change in the environment, we are applying some sort of a processing which is a stress to the bug. They start secreting certain molecules which are like acyl homoserine lactones, and these attract more bacteria of the same kind or of a different species to come near them and settle along with them. Okay, so the initially stuck bacteria, they invite more bacteria and they keep on, they stick with each other and they attach to the surface in a kind of an irreversible phase. Okay, and then they keep on growing in this attached phase to mature and form a biofilm, which is like a community of cells settled sticking onto a surface so that they can resist the treatment we are giving. So we are adding a chemical, or we are adding certain pH changes or temperature or pressure, whatever. So the bacteria, when they are growing together, they have like a limited penetration of the stress we are in putting in. Okay, there is a limited penetration, and there are also mechanisms to neutralize that stress. And then that is how they can actually succeed in resisting the environmental pressure we have induced. And then gradually, when the size of the film has really increased, they may actually they may get plucked off and they disperse to new locations. And this is a continuous source of basically contamination of the foodstuffs in the food industry. So biofilms, if we have to define them, they are layers of bacteria that attach to the surfaces and to one another. with the help of a polymeric material, like which is the exopolysaccharide, which helps in trapping other bacteria, debris, and nutrients, and provide a blanket to the cells. On the right hand side here, we have different phases of growth of a biofilm. And that is how at initial attachment, then growth, and then really becoming a three-dimensional structure. And this is what is commonly seen in the industries. And what are the advantage that biofilms offers to the bug? It basically provides it protection against antimicrobials and other processing strategies. It enables the bacteria to persist, concentrate nutrients. It is also a ground of genetic exchange where genetic material can be actually exchanged easily and new resistance can be acquired by the species which are forming the biofilms. And there are a lot of cross species interactions, and evolution of the strains are a common thing which is seen in the biofilms if we study them for a longer period of time. What is the concern to the food industry? Of course, it is a major cause of concern to the food safety that if the biofilm forming strains are pathogenic, they may spread the disease. They're resistant to the antimicrobial. So what do? How do we get rid of the bacteria if they are already showing resistance? So it reduces the shelf life of the food item, thereby inducing spoilages. It impairs the heat transfer and are a major economic concern. Okay. I'll be talking more of that a little in a few slides from now. And biofilms, basically, I've already actually discussed this, so I will just give you an overview. What is the composition of the biofilms? What is it composed of? There is slime, so a lot of polysaccharides are there, which are present extraneously outside the cells. There are proteins, lipids, DNA. There are also inorganic compounds and nutrients which are trapped in this matrix to enable that there are cells which keep on growing. So that's the basic composition of the biofilms. And I've already covered this. What are the survival advantages of the biofilms? So when do the microbes decide to form the biofilms? It's mainly when they come in contact with the specific or a non-specific attachment sites or surface. So we can say it as a food material wherein they may stick. There are environmental signals, as I just told you. It can be processing the signals or any other kind of stress, nutrient starvation, etc., which may serve as the environmental cues to trigger this biofilm to basically turn on the genes for biofilm formation in the bacteria, and then exposure to sub-inhibitory concentrations of antimicrobials that we are doing. So this is what is these any one of these can serve as the signals for trigger biofilm formation in bacteria. So what is the impact of biofilms in our lives? Biofilms are found everywhere. It can be on our teeth, which is known as the plaque. Streptococcus mutans grows as biofilms, forming plaque on our teeth. It's very difficult to get rid of it once it starts. And then even in the oil industry and the cooling water supplies or even the ship hulls, the films cause a lot of problem. When they grow, they are going to basically erode and basically corrosion of the materials they can easily cause in the food processing industries. They can lead to spoilages and contaminations and even transmission of diseases in the medical implants as well as in the paper and pulp industry. So I have few figures here where biofilms formation, the prosthetic joints is a major concern. It grows even in the heart valves which are fitted in and cause recurrent infection in the individuals who have undergone these transplants. I've shown the image of the plaques. There is a bug known as Pseudomonas aeruginosa, which causes cystic fibrosis and it clogs the airways, leading to cystic fibrosis. It's really a deadly disease wherein the person who's suffering from these, who is infected with the biofilm forming Pseudomonas, they suffer from lung diseases and they don't survive for a very long period of time. It's very difficult to get rid of this infection and the medications don't work because of the resistance shown by the bug. And interestingly, even on the lenses, contact lenses, biofilms are formed by certain bugs. So in our homes, you all might have witnessed such kind of things which are growing. So in the bathroom flush systems or even the kitchen sinks, washing machines, on the walls, If you open the pipelines, you'll be seeing growth of microbes. And even if you clean it, initially it'll look very clean, and then within a few days again, it comes over. So these are all biofilms. And in the biomedical industries, it's on the catheters and everywhere in the dialysis units that the bacteria may grow as biofilms. In the food industries, the basically okay, this is basically not for in general industries in the oil refineries and all. They're going to grow on the pipelines, and it appears as a corrosion, but if we analyze them microbiologically, we see that a lot of films of bacteria have grown, and it's very difficult to get rid of it, and it causes erosion of the surface, corrosion of the surfaces. And in the food industries, they grow on turbines, they grow in the drain pipes, they grow on the surface of the food materials, they grow on the heat exchangers, affecting the efficiency of of the process. Okay, there is very little transmission of heat if it happens in the pasteurization pipelines and all. So you can imagine that there is insufficient transfer of heat, and the bacteria would probably be contaminating the entire food material, as well as inefficient process is there. So it's a major problem to the industry. Okay. So, a few examples in the food industry, they're a cause of continuous recontamination of foods, okay? And especially because the equipment, the processing equipment is inhabited by these persistent bugs. The removal of the persistent bacteria is definitely reduces the productivity, and we are using very harsh chemicals and treatments to treat these biofilms and to get rid of the biofilms from the industry. So it's basically those harsh chemicals are environmental safety concerns, and even they can be toxic to us if we happen to consume them, and if they are not removed properly from the surfaces which were treated. And there's a basically estimation that the food poisoning which are occurring because of the biofilm forming bacteria which are present in the food processing units, they can cost over 6.5 to 34.9 billion dollars annual cost to the industry. Okay, and this is a little old data. It is still as high as it was reported earlier. And ultimately, what happens is that the products are contaminated, and it may lead to recalls, resulting in really huge and expensive losses to the food companies. And to the food safety, the impact of biofilms is that it leads to product contaminations. As I just mentioned it, if the cells are released, as I told you earlier also, so that if the biofilm forming bacteria were pathogenic, they can lead to a major foodborne illness or an outbreak. And there's an example from United States that in April to July 2008, there were more than 1,300 people across 43 states which are infected with salmonella poisoning from tomatoes. And in morning, we know that Dr. Govind gave a very good presentation on salmonella presence in tomatoes. So it's a major concern there. And it was because the washing water, which was used, was contaminated, and the biofilms were formed on the tomatoes. And the impact it had was that the restaurants for several months could not serve tomatoes on their food products. And we know how important tomatoes are. They're a part of sauces, they're consumed raw, they're part of sandwiches, burgers, you name it, and it's all there. So it had a huge impact. And this is a very small example which I'm giving you. So imagine if there is dairy products, there is meat products, there are n number of products when biofilms microbial biofilms can form. So impact on the food safety is huge and immense. So the routine biofilm interventions or control strategies are you are doing a good CIP treatment and disinfection regimes. After every single batch of manufacturing, the surfaces or the pipelines are treated with hot water, alkalis, and certain other antimicrobials. But there is a problem actually. First of all, it doesn't ensure 100% removal of the bug. Okay, if the bug has the capability of forming biofilms, there will be few cells which might tolerate the concentrations which are being used. Okay, and over a period of time, they form biofilms, and they are basically diagnosed very late when the damage is already done. And another problem is that these alkali and the antimicrobials which are being used are toxic and unsafe. And as I told you, they are environmental pollutants, and they also induce more resistance amongst the bacteria. So we are actually having a scarcity of antimicrobials which are very safe for getting rid of the biofilms, and that's how where work is going on. And these are few more novel strategies which have come up in literature. There are surfactants and biosurfactants, surface coating, adhesion molecules, etc., which are used for basically getting rid of the biofilm forming bacteria. And now I'll just move to some more exciting research. So, where do we look for safer alternatives? Is we go back to the nature. Okay, nature has a lot of molecules to offer, which have a natural way of preventing the growth of bacteria, which are forming biofilms. So, plants, animals, microbes, each one of them have some metabolites or some essential components which will probably have a good antimicrobial effect against the resistant bugs. And we may try that application directly on the food materials. We can incorporate them as edible biofilms or coating, and we may even deliver them as nanomaterials and ensure food safety. And the advantage is that they have been used for years for treatment of diseases. They are safe. They can be used as nutraceuticals, preservatives, as well as extend the shelf life. There are no side effects. It's very cost effective. It's very readily available in the nature. It's all there in the nature, and it's biodegradable. So these are the kind of advantages. And what has to be done is, if you have any kind of a prospective candidate present, say for example in a plant, it can be a fruit, it can be leaf, stem, roots, anything. You may prepare a kind of an extract from it. And yesterday we had a very good presentation by Dr. S. Nayak, who basically presented his work on solvent extraction strategies of bioactive compounds. So those kinds of extraction techniques can be done to isolate the active ingredients. Then some bioassays can be done to basically screen out for the bioactivity. In our case, it will be the biofilm formation. So if there is an extract showing a positive biofilm inhibitory properties, there will be a good way of screening for this thing for further going down line and purifying the components, active ingredients, elucidating their structures and understanding their mechanisms. How do they kill? And then we may even develop them in the laboratory level to get more highly efficient, antimicrobials. So the various ways in which they usually are inhibiting is either by inhibiting the cell wall formation, by stopping the toxin formation, inhibiting the energy synthesis or the ATP synthesis machinery, quorum sensing, etc. Okay, there are n number of ways, and most of the like antimicrobials are of natural sources are the ones which are producing reactive oxygen and reactive nitrogen species. So they are really high they are high oxidizing agents. Okay, so in our lab, what we have been doing is we are following a kind of a in silico approach or a molecular docking or a bioinformatic approach to rationalize and identify chemical ligands from the food from the plants, which would be effective in killing biofilm forming foodborne pathogens. And we do a kind of a in vitro analysis to find the dose of their efficacy. We have developed certain bionanocomposites, and we try to see what is the effect at the genetic as well as at the physiological phenotypic levels. We determine its toxicity, and we have developed it as coatings and active packaging. So I am just going to discuss one of the examples. We have many more, but one of the thematic examples would definitely help the audience in understanding how the work is done and what is the way to go. So we started with isolating and identifying foodborne pathogens from present in Indian dairy and meat samples, and we isolated E. coli, Salmonella, and Listeria also from the food materials, and we found that majorly in the dairy and the dairy as well as in the meat industry, there were a lot of samples. More than 50% samples were positive for presence of these bacteria. Okay, and this is then we identified them further by biochemical and molecular methods, and we found that the isolates were of E. coli. We have published it in International Journal of Food Microbiology, where a new strain of Escherichia coli has been isolated from meat, and it is a biofilm former. Then what we did was we screened for the biofilm forming activity of these isolates using the crystal violet assay and adherence to the surfaces. Okay, and we found that more than 47% of the population which were isolated from the meat and the dairy was moderate, could form moderate to strong biofilms. So they are all capable, almost 50% of them are capable of forming the biofilms. And then we also mapped its biofilm forming rates, etc. And we could see that over a period of very early on, from 2 hours onwards till 4 days, it could form very good biofilms. We further characterized the biofilm forming potential of one of the strong biofilm formers. And so EMC17 is a strong biofilm former. And we saw its quorum sensing production activity as well as its efficiency in exopolysaccharide production. And we found that the EMC17 strain of E. coli could really have a good quorum sensing signal generated over an early time point from 2 hours to 8 hours, because quorum sensing or the signal transduction for starting the biofilm formation is an early time point phenomena. So we did see that as compared to the weak biofilm forming strain, a very strong signal is generated by the bugs. And then over a period of 96 hours, since we knew that from 1 day to 4 days the biofilm formation is increasing, we mapped out what was the kind of rate of EPS production. And there was a huge production of exopolysaccharide by the strain, vis-a-vis a weak biofilm former did not really generate a good EPS. So these were all the ways we characterized the strain we isolated, and here is an actual picture of the EMC17 strain over a period of time to just show what are the various stages of biofilm formation of this isolate. Okay, so after this, we did an in silico screening of phytochemicals. And in order to do so, there are a couple of things which are required. So we selected what should be the microbial target for for targeting the biofilms. We identified certain receptor genes from the literature which are responsible or which play a very important role in biofilm formation. Each of them is having some specific role, right from production of the quorum sensing signal to the production of the EPS, etc. So we basically identified certain receptors and pulled down their structures from the literature. Parallelly, what we did was we basically selected an Indian medicinal plants library to select for or identify the ligands to these receptors, right? So the impact library, which has a database of almost 10,000 phytochemicals from the Indian medicinal plants, we were able to screen out the library, and we identified almost 140 compounds randomly were selected based upon their classes as well as their easy availability to absolutely mix and match to basically target the receptors we had identified on the bacteria. And how it goes is that we download the structures of the phytochemicals, prospective phytochemicals from the impact library, which we have chosen. We convert it into another format, and then parallelly we have downloaded the structure, the receptor, and we basically do a molecular docking, or we try to see how good they fit in or interact with each other by running a simulation and looking at the free energies. So higher the match, so lower is the E value of the thing. So For screening of the phytochemicals, we basically measure the E value predicted by the software. We looked at the commercial availability of the material phytochemical, what is the class and diversity, and what are the economic values, how easily it can be actually procured, right? So from the in silico studies, we basically were able to list down the 10 phytochemicals from their various sources which are listed here against these targets of the biofilm formation as well as virulence found in E. coli. And we selected quercetin because it was showing a very good activity. So when we did the preliminary screening of the selected 10 selected phytochemicals, we saw that there was a good amount of inhibition of biofilms by almost all of them: curcumin, quercetin, menthol, piperin, gallic acid, eugenol, and ferulic acid. All of them at concentration somewhere around, I think, 1 mg per ml were able to inhibit the biofilm formation anywhere from 80% to almost completely. And then it was more or less concentration dependent. Okay. And in order to confirm our results, we also did certain microscopies. And in the fluorescent microscopy, so you see this is an untreated biofilm. And upon treatment with curcumin, and each one of this is a sub-inhibitory concentration. So after we had calculated the MICs, after we had determined the minimum inhibitory concentrations, we took the MIC 50, where 50% of the cells will survive, so that we can visualize them under the microscope. And we saw the efficacy against the biofilm of these phytochemicals against the biofilms. From this, we selected quercetin as one of the molecules because it showed a very good activity. It also showed very good activity against both biofilm as well as the planktonic or the free floating state. But there was a problem which is very commonly reported and which has been a challenge to meet. Most of the phytochemicals, they are very unstable. Okay. And stability is a very important thing to maintain the biological activity. Okay. Because the instability, what happens is they on exposure to temperature, which is a common thing used in the food industry, exposure to light, or exposure to oxygen. Many of these phytochemicals lose their biological activity or bioactivity. Okay, and what should be done? So if we add it to a food packaging material, or if we coat it onto the surfaces, they would gradually lose their activity, and it will not provide a prolonged efficacy against the biofilm forming bacteria. So what we did was we tried to have a kind of development. We used the nanotechnology to develop a quercetin loaded nanomaterial. Okay, and this work in this work we chose mesoporous silica as the kind of loading material onto which quercetin was impregnated. Okay, so this mesoporous silica impregnated with quercetin was then tested for its efficacy to work against the biofilms or for the isolate which we were working on. So this is like a general diagram depicting what was done. So synthesis and characterization of the mesoporous silica loaded with quercetin was done. There were X-ray diffractions, SEM and TEM images, all showing that yes, quercetin is completely loaded. And then what we did was we did the biological activity of this against. So we try to analyze what are the minimum inhibitory concentration of the loaded bionanocomposite, and we saw that at 100 micrograms per microliter it was showing a very good activity, and then gradually it was a concentration dependent thing. So it was very promising because it could actually kill the biofilm formers, as is also shown in this fluorescent microscopy image. And we have further actually done studies to determine how this killing is being done. So we found that it was majorly by the generation of oxidizing, reactive oxygen species that quercetin loaded bionanocomposites work. So we saw that there is an assay, which is the Dcfda assay, wherein a dye is used to indicate for generation of the reactive oxygen species, which indicated that there is a good amount of ROS generation. So a reduction in the fluorescence by the quercetin and the nanocomposite quercetin, like bio-nanocomposite, clearly indicated that there was a generation of reactive oxygen species vis-a-vis the carrier moieties. Okay, so it is a clear cut, and then I haven't included it here, but we also saw that the treated cells, they had damage to their cell wall, and they there was a lot of protein leakage and sugar leakage by the cells, which we have tested. I haven't included it here though. And after all the kinds of studies, we also did certain gene expression studies to actually analyze what kind of genes, which are what is the impact of quercetin and nanocomposite quercetin on the gene expression profiles of biofilm forming genes. That data is not a part of the slide though, and we found that major genes which are involved in quorum sensing, signal production, as well as in EPS production, are down regulated when it is treated with these materials. So at the genetic level also we saw a confirmation, and we also have a confirmation here at the phenotypic level, right? So after doing all this, how do we use it, right? We did certain stability studies. So we saw that even after exposure to light and exposure to oxygen, the bio nanocomposite was very stable for a period of almost 2 months, right? And that means that that's very promising. So if for a period of 2 months even light exposure and oxygen exposure is not eroding the activity of this quercetin binanocomposite, we should be able to use it. And what we did was we tried to coat it onto stainless steel surfaces, because in most of the industry stainless steel is the material which is used in on processing surfaces. That's the material. So we had stainless steel coupons of 316, and we coated it with quercetin, and we had a control which is uncoated, right? And this is bio-nanocomposite quercetin. And then we basically dipped it in a bacterial culture, and we left it for a period of 24 hours for biofilms to form. And then we observed the amount of biofilm formation microscopically, as well as we enumerated the bacteria which are sticking. And we saw that there was a huge reduction in biofilm formation on like quercetin coated stainless steel coupons, right? So this is like a proof of what we have done. We have actually seen that quercetin by a nanocomposite is able to prevent the formation of biofilms on stainless steel coupons, and this material is very, very stable. And what is the way forward? We also are now trying to incorporate it, the same material, in food packaging films, so as to provide a protective layer around the food. So the food can be actually protected by either dipping it or encasing it in a surface which is or having huge quantities, or whatever the minimum inhibitory concentrations indicate those quantities of the biomaterial. And gradually we may even have intermittent layers to protect the direct interaction, so as to prevent any kind of sensory alterations to the food. So there is a good way of generating films around the food or packaging around the food, which we are exploring now. And these natural phytochemicals can actually be used in this manner to prevent the growth of biofilms and prevent spoilage of food materials. So the concept here was to show it as surface coatings, and here we want to basically try for antimicrobial packagings. I would also like to tell you that besides the phytochemicals like quercetin, curcumin, etc., we have also worked with vitamins. And there's another published literature we published in Food Control this year. Vitamin C, which is
Seems that the speaker has some technical glitch, so it will be rectified soon and then the presentation will continue at the earliest. The speaker will be joining us very shortly in 2 minutes. So as I told, there was a technical glitch and then it's been rectified and then the speaker will be available in 2 minutes.
Hello, everyone. Am I audible?
Yes, ma'am, you're audible. You can
I
continue.
am so sorry. I think there was some sort of a internet issue here at my end. So I just concluding. I'll just put on my slide here again and conclude the presentation. I was almost at the conclusion. That's the worst thing which can happen, but that's how technology is. Sometimes glitches do happen. So just 1 second. Um, So, in order to conclude my talk, we have discussed what are the mechanisms of biofilm formation and how important and relevant they are to the food industry and food safety. And we've also shown you how natural phytochemicals can actually serve as very safe candidates for curbing and curing biofilms. So they are very promising candidates, and nanotechnological interventions can actually help us in attaining stable moieties and achieve better applications in the food industry. And it will be very unfair if I do not acknowledge the people who have been the backbone of this work. First of all, the funding of this project by SERB grant and the Ministry of Food Processing Industries helped us in getting the funds for conducting this work. And the main work has been done by Mr. Shiva Prasad DP, who's also a faculty at Vignan Foundation here. So he's the one who has worked really day and night and very hard to complete this work. And besides that, molecular docking and other stuff which is going on the lab is being taken up by my PhD scholars, Mr. Dinesh Bhardwaj and Ms. Akanksha Joshi. And I would also like to thank my collaborators, Dr. Divya Sachdev, who's a nanotechnologist who helped us in getting the bio-nano composite done. And we are working together even in many other works on diagnosis via nanotechnology, right? And Dr. Renu Pasricha, who's a scientist at the New York University, Abu Dhabi, who has helped us with the microscopy studies. So I would like to thank everyone and for your patient listening. And I would once again like to thank the organizers for having invited me and giving me a platform to share my work. I would take any questions if there are. I would be happy to answer them. Thank you so much.
Thank you, ma'am. Participants, if you have any kind of queries, please feel free to ask or you can post it in the chat box. Ma'am, there is one question from a participant. How can we curb the vitamins? I mean, how vitamins are effective in curbing the biofilm?
Yeah, so as I just the work has not been discussed here. I did not portray that work here. We have seen that vitamins like vitamin C, it's a very good antioxidant as well as pro-oxidant, right? So it generates a very high amount of reactive oxygen species, and by its ROS generation, it has its antimicrobial activity. So it was just like a kind of exploratory project that we had undertaken, and we saw that at certain concentrations, vitamin C showed a very good activity in eliminating biofilm formation. It stops the quorum sensing, it stops the EPS production, it also stops the ability of the bacteria to adhere to surfaces. So we just published it this year in food chemistry, in the journal Food Chemistry, right? And for more details, maybe what I can do is they can contact me. I can share the manuscript with them. So majorly by the ROS generation, vitamin C can very effectively have an antimicrobial effect, right? And even during the corona time, okay, people have been advocating a lot of vitamins, not only to boost your immunity, but also because they have a very good antimicrobial activities. So they are like dual moieties, okay? On one hand, it's an antioxidant, on the other hand, it's a pro-oxidant, depending on the concentrations in which we are using.
Thank you, ma'am. There is another question. Is quercetin nanocomposite applicable in industrial level? And if so, is it not affected by cleaning and sanitation method? And will there be any effect of nanocomposite on food products?
So basically, see, any of these phytochemicals, quercetin is just one of the examples we are trying to show here. Any of these natural compounds which we are trying to advocate, um, they are the ones which we want to apply directly on the food material, okay? Onto the surfaces, like say stainless steel coupon is one of the ways, and that also we tried because we were using a matrix, a nano matrix to deliver it. But of course, it has to be a regular practice. We are yet to evaluate it, how frequent applications of this nano composite should be required to prevent the formation of biofilms. So since we did it only for 24 hours, we'll have to do really a time planned manner of the study to see what is the kind of like how effective it is for what duration of time, when a reapplication of the readministration of this quercetin has to be done onto the surface to prevent any biofilm formation, right? So all those studies will have to be done. Of course, if it is incorporated in the food material or in the packaging material, so once it's packed and once it's sealed, we expect that it should be able to really extend the shelf life and kill it. It not only extends the shelf life, it also kills the pathogens. So if there was say 1,000 bacteria or a million bacteria over a period of time, due to the interaction, the slow release of quercetin from the food from the packaging material, we expect that there would be no biofilm formation and there would be absolute sort of sanitation of the product for a given period of time. Okay, so for application onto the food processing surfaces, a recurrent application and a timely time regimes, it would, we wish that we can replace all chemical sanitizers. Okay, any kind of harsh chemicals need to be replaced by these safe chemicals now. So that is what our aim is.
Thank you, ma'am.
Yeah.
There is another question. Are there any methods to prevent vegetables and fruits from biofilm spoilage kept outside the refrigerator?
Uh, so basically, biofilms, as far as when we talk about the biofilm formation on fruits and vegetables, or even on meat and dairy products, any kind of a product, definitely if we are able to reduce the load of the biofilm formers or toward any kind of a microbial population on the surface, we can keep them, we can extend their shelf life. But see, we must understand that spoilage, food spoilage is not only microbial, but it can also be chemical. Okay, the ripening of the food material and the enzymes which are involved, which are present naturally in the food item. So these phytochemicals that we are targeting or which we claim are only going to work against the microorganisms. We haven't really seen how effective they are in reducing the enzymatic activity which is naturally there in the food. Okay, so definitely as far as the microbiological safety of the fruits and vegetables is concerned, we do expect that we should be able to reduce the load of the microorganisms and store it on the shelf for a longer period of time. But we cannot really claim how effective it is in reducing uh, the enzyme and the other kinds of activity which is within the product.
Can bioenzymes help in inhibiting the growth of biofilms?
Uh, So, as far as the enzymes and all are concerned, I'm sure there would be work going on. I haven't really checked on that. I'm not an expert to comment on that. But I'm pretty sure there are enzymes. Say, for example, if I have to make a guess, then any kind of an enzyme which is going to break the exopolysaccharide EPS, you know, should be working very well against the biofilms, right? But they can be used as treatments to break the biofilms. Okay, but I think more literature search is required to answer this question. I will not really be able to answer it correctly, but yes, enzymes can be used, those which can break down the complex EPS. But remember that there are, yes, yeah, there is one called dispersin, and that dispersin is able to break down the biofilms. But remember that these enzymes are really expensive ones. We should have a strategy to manufacture them at a high level. And secondly, enzymes are very sensitive molecules in themselves. They are sensitive to pH, they are sensitive to temperature, they are sensitive to many other organic materials. So if we want to use a bioenzyme, we need to ensure that depends on what condition the food product is being stored and processed and and delivered. So all those things, I don't think enzymes can be the best ways of working against biofilms for the sensitivity they have in themselves,
Okay. Thank you, ma'am.
right?
There is one more
Yeah.
question. Can antimicrobial bio nanocomposites can be applied for cut fruits and vegetables?
Yeah, I think I've answered this question. That any kind of these, see, we have to determine if we wish to apply something on food material. Safety is something which we need to ensure. Okay, so after ascertaining its safety, that it is non-toxic, we can, but we haven't really tried it on cut fruits and vegetables. But we are sure. See, remember that these nanomaterials have to be safe first of all. Secondly, the phytochemicals are safe in their own ways. At the doses that we have tried to see, they're very stable, and they're very safe. The concentrations in which we have used it is within the safety limits of their application. Secondly, um, they are having their own health benefits. We know that you must have seen that we've used one example of curcumin. We are also working on curcumin. So curcumin, which is present in turmeric, has its own anti-cancer effects. They are used as therapeutics. Okay, even quercetin is used as a therapeutic. So by virtue of their therapeutic value, there is, I don't see a reason why it cannot be applied on any of any of food materials, be it cut fruits or intact ones, or even any kind of processed meats and dairy. So yes, they should be, they can, and but we need to, at least in my lab, we need to actually do further studies to try it on various kinds of food materials. At present, we have tried it in dairy industry, in paneer, And we're also trying to now move on and test it in meat products and meat safety.
Thank you, ma'am. That is the end of all the questions. And I please
Thank
request
you.
Mr. Prasad to conclude the session.
Thank you, ma'am, for such an interesting and very informative presentation and imparting all the participants with the knowledge of biofilm and its significance as far as food industry is considered, and use of natural antimicrobials and their nanocomposites to curb the biofilm formation from the food processing surfaces. Thank you very much for sparing your valuable time and then speaking to us. Thank you very much, ma'am.
Thank you. Thank you so much.
Thank you, ma'am.
In conclusion of this particular technical session, I would like to thank all the keynote speakers, both national as well as international speakers, for agreeing to speak to us by sparing their valuable time in their busy schedule. Thank you very much to all the participants. And I would also like to mention that the next session would commence exactly at 1 p.m. Thank you all.
Okay, participants, please be back at 1 o'clock. We will be starting the session at 1. It's going to be lunch break now.